This method can help answer key questions in the fields of cell biology and immunology, such as how are signaling pathways affected by different stimuli or drugs. The main advantage of this technique is that it allows you to perform single-cell signaling analysis in a medium to high-throughput fashion.
After resuspending the cells in supplemented RPMI 1640 medium, transfer the required amount of cell suspension to the wells of a 96-well V-bottom plate. Transfer the plate to a preheated 37 degrees Celsius water bath and allow it to rest there for 10 minutes. Then add 60 microliters of fixed buffer 1 to each well of a new 96-well plate to set up the fixed plate. Place this plate in the 37 degrees Celsius water bath. Transfer a 50 microliter control sample to the plate and mix by pipetting up and down. Optionally, add 10 microliters per milliliter anti-IgM to the cells to start the simulation time course, and mix by pipetting up and down. Transfer a 50 microliter sample to the fixed plate at each time point, mixing each sample as it is added. After the last sample is added, leave the fixed plate in the water bath for 10 minutes. First, wash the cells 3 times with PBS. Centrifuge the plate at 500.